Alex Baina here and welcome to Biochemistry, and in today's episode we'll be talking about enzyme. So what is an enzyme? What are the functions of enzyme in our body? And how are enzymes inhibited or controlled? Let us recall about proteins, one of the biological molecules in our body. Now, proteins, the different functions are structural component. An example to this is the protein called actin. Actin actually forms the microfilaments in our cytoskeleton. So these are the cytoskeletons of a cell, and these cytoskeletons are called microfilaments and are made from actin. Another function of proteins in our body is antibody. An example to this is immunoglobulin. This is the structure of an immunoglobulin. Next, protein can also function as a transport and a storage in our body. An example is ferritin. Ferritin actually contains iron. A deficiency of ferritin indicates anemia. So this is the structure of ferritin, and these molecules here are iron. Iron are actually stored and transported via ferritin. And a person with anemia can have the symptoms of tiredness, weakness, pale skin, irregular heartbeat, shortness of breath, chest pain, dizziness, cold hands and feet, and also headache. Now, proteins can also function as a messenger. An example to this one is growth hormone. Growth hormones. Growth hormones control your body's growth. It is made by the pituitary gland, and it increases muscle mass and decreases body fat. And finally, the function of protein can also be catalysis of biomolecular processes, and that will be handled by enzyme. And this will be our lesson for this segment of biochemistry. So, enzymes. What are enzymes and what are the functions of an enzyme? Enzyme structure are mostly globular proteins, meaning they are in the form of tertiary or quaternary structure. These are the different representations of a globular structure of a protein, specifically an enzyme. Now this cavity over here is what we call the active site of an enzyme, and this one is the attached substrate of an enzyme. So the substrate is the one that we will convert into the product, or what we call biological process. As discussed already in the previous slide, an enzyme has an active site. The enzyme active site is divided into, the first is binding site. The binding site of an enzyme is where the substrate can bind to the enzyme itself. This is the illustration of an active site and a substrate. So the enzyme itself has a cavity where the substrate can attach. Another division of the enzyme binding site is called the catalytic site. Basically, this catalytic site is just found in the enzyme active site. The catalytic site of an enzyme or an enzyme binding site performs the catalytic action of an enzyme. The catalytic action of an enzyme is what produces the product. So how small is that enzyme active site? It is only less than 5% of the total enzyme surface area. So again, this enzyme active site will bind to the substrate, or we also call it the reactant. As the substrate or the reactant binds into the enzyme active site, this will form what we call the enzyme substrate complex. As the substrate binds into the enzyme active site, this will create what we call the enzyme substrate complex. In other words, that the enzyme has already the substrate that is attached to it. And later on, that will be processed that will produce the products later. The enzyme active site will also bind to a cofactor or cofactors. These cofactors will help the enzyme catalyze the reaction. Cofactors are mostly made of vitamins. Enzyme cofactors. These are chemicals that assist enzymes during the catalysis of reactions. Cofactors can be metals or small organic molecules. In this illustration, this is a cofactor. It's a vitamin or a mineral, and this is an enzyme. So an enzyme has an active site. In this case, it is an incomplete active site. So once the cofactor is attached to the enzyme active site, the substrate is now able to bind into the new configuration of the enzyme active site. And that is how the enzyme will later on process the substrate to form the new products. Cofactors can be classified as a prosthetic group that is bound tightly to the enzyme. Prosthetic groups are non-protein components that attach mostly to proteins. They are compounds that are bounded to our enzymes. This is the enzyme, and this is the active site configuration that is created between the enzyme and the prosthetic group. Another classification of a cofactor is a coenzyme. Coenzymes are bounded loosely with the enzyme, unlike the prosthetic groups that they are bounded tightly to the enzyme. Coenzymes are organic molecules that bind loosely to the active site of an enzyme, and they also aid in the substrate recruitment. In the illustration shown, coenzymes or a coenzyme is already attached to the active site of an enzyme. Together with the cofactor, coenzyme will also generate the new configuration of the catalytic site of this new enzyme. Characteristics of an enzyme. Enzyme catalyzes more than 5,000 biochemical reaction types. An example to these biochemical reaction types are digestive enzymes such as trypsin and pepsin. During the biochemical reaction, the enzyme has no effect to the product of the reaction made. Let us again remember that an enzyme is made from proteins. That means enzymes are sensitive to the change in temperature. In other words, enzyme operates in what we call the optimum working temperature. This is a graph that is showing the optimum working temperature of an enzyme, that when an enzyme will be operating at a much lower temperature than the optimum working temperature of an enzyme, the enzyme becomes inactive. While if the enzyme is exposed to a higher temperature, the enzyme itself denaturizes. Enzymes are also sensitive in pH change. Thus, an enzyme is also or will be working with what we call the optimum pH scale. For example, the optimal pH for pepsin, the stomach enzyme, is a little bit above pH of 2 but is not more than the pH of 3. However, For trypsin, the intestinal enzyme is working between the pH of 8 and 9, that is 8.5. It should not be lower than 8 or more than 9, because if this happens, the enzyme probably will be denaturized. Enzymes are also sensitive to the substrate concentration, or the amount of substrate that it will actually process for biochemical catalysis. So, enzymes will work best if there is plenty of substrate. So thus the rate of enzyme activity also follows. Enzymes are highly specific, meaning enzymes only catalyzes a specific type of substrate. What is an enzyme turnover number? The enzyme turnover number is actually the number of substrate molecules that can be transformed by the enzyme per minute. An example to this is the enzyme catalase. It can transform about 6 times 10 raised to the power of 6 molecules per minute. Now, enzymes are synthesized or created in the ribosomes. Specifically, the ribosomes that are attached to the endoplasmic reticulum. These are the rough endoplasmic reticulum. This is an image of the rough endoplasmic reticulum. So these are the ribosomes that are attached to the endoplasmic reticulum that is found in our cell. There are two types of enzymes. The first type is extracellular enzyme. The extracellular enzymes are secreted from the cell for external work. An example to this are the digestive enzymes. So enzymes for external use are actually created in our cells, and they are packed using the Golgi apparatus, and they are excreted outside of the cell. The other type of enzyme is called the intracellular enzyme. The intracellular enzymes are retained inside the cell for use. An example to these are the enzymes involved in the oxidation reduction inside the mitochondria, also known as the oxidoreductase enzyme. The oxidoreductase enzyme catalyzes biological oxidation. When a cofactor is removed from an enzyme, this is called an apoenzyme. An apoenzyme is an inactive type or form of an enzyme. However, when an apoenzyme has the cofactor attached to it, it is called an active enzyme. This is also known as holoenzyme. This is an apoenzyme plus a cofactor. This is an example of a holoenzyme. This type of enzyme is the active form of enzyme. If we remove the cofactor, this will be called an apoenzyme. Now, do you ever wonder how enzymes are named after? So actually, the naming system of an enzyme is basically called Enzyme Nomenclature, or also known as the naming system of an enzyme. There are two naming systems that are being adopted in an enzyme nomenclature. The one is that ends with IN, such as renin, trypsin, and pepsin. So Pepsi-Cola, the competitor of Coca-Cola, was named after the digestive enzyme pepsin, because it was believed before that Pepsi is a health drink that helps or aids in the digestion. The other type is it is based from the type of substrate that the enzyme catalyzes. This is adding a suffix ase. For example, hydrolysis enzyme is now called a hydrolase. meaning to say that hydrolase is an enzyme that is used for the process of hydrolysis. The enzyme classification is developed by the International Enzyme Commission, and this classification is based on the types of reaction that is catalyzed by an enzyme. The following are the 6 classifications of enzyme. The oxidoreductase. This oxidoreductase is for the reduction-oxidation reaction. An example to this is the lactate dehydrogenase enzyme. Another classification is called ligases. This is for the synthesis of new covalent bond. An example to this is the RNA polymerase enzyme. Next is lyases. This is for the cleavage without the use of water. An example to this is the fumarase enzyme. Then we have the transferases enzyme. This is for the transfer of chemical groups. An example to this is the hexokinase enzyme. Next is hydrolases. This is intended for the cleavage of bond with the use of water. An example to this is the lysomase enzyme. And finally, the isomerases. This is for the transfer of intermolecular group. An example to this is the triose phosphate isomerase enzyme. Enzyme action mechanism. There are 2 explanation perspectives about the enzyme action mechanism, and the first one is thermodynamics changes. It is said that a chemical reaction have energy barrier between the reactants and the product. That energy barrier requires the activational barrier for the product to occur spontaneously. And that activational barrier or the energy barrier can be lowered only by the use or by the means of an enzyme. There are 2 illustrations in here. The other one has the normal activation energy, and this is the energy barrier that will be lowered down by the use of an enzyme. So our reactant will require a very high energy barrier in order for the reactant to produce spontaneously the product. But with the use of an enzyme, that normal activation energy barrier can be lowered. This is called the activation energy that is lowered by the use of an enzyme. In other words, the reaction will only require a very small amount of energy but still produce the same product. And so with that, we will be looking at the different processes at the active site that is happening of an enzyme in its catalytic conversion. And this is the other perspective. The first enzyme action mechanism is called the acid-base catalysis. This type of reaction requires the oxidoreductase enzyme. This lowers the activation energy barrier or speeds up the reaction by the addition of acid or a base. Acid-base catalysis of an enzyme. The chemical reaction is accelerated by the addition of an acid or the addition of a base. Proton transfer is the most common reaction that the enzyme performs. An example here is the serine protease. So the proton is now transferred to this element. So H transferred to B, therefore it changes the configuration and releasing the product. The other enzyme action mechanism is the type called covalent catalysis. This is where the enzyme forms a covalent linkage. This covalent linkage can lower the activation energy and thus will catalyze the specific reaction and release the product. This is how the covalent catalysis looks like. Here we have the enzyme and the enzyme active site that has the catalytic site on it, and this is the substrate. And so there will now be a form of covalent intermediate between the product and at the same time the enzyme catalytic site. And with this, it releases the other product or the portion of the substrate and eventually releases the two products. The other enzyme action mechanism is called catalysis by strain. This enzyme mechanism type utilizes the lyase type of enzyme. Catalysis by strain causes the structure to reorient, or structure reorientation happens in here. This creates an unstable bond, unsnaps, and then this will also cause to release the product into the environment or cellular environment. So this is now the action of enzyme catalysis by strain. First, the enzyme accepts the substrate into the active site, and therefore there will be an orientation and eventually snaps and produce the product into the environment. And finally, catalysis by proximity. This type of enzyme action mechanism releases high substrate concentration, where it will create the ideal substrate orientation that will finally create the product. In here, the substrates are held close in space and in the correct orientation. In other words, held close is proximity. Catalysis by proximity will result into a more efficient reaction pathway, and thus, with this, we'll already release the product. Enzyme substrate model. These are the types or models that are hypothesized as how we see the schematic diagram of the substrate and our enzyme. Now we are going to look at the first type, which is the lock and key model. This is proposed by Emil Fischer in 1894. This lock and key model suggests that enzyme substrate interaction possesses a specific complementary geometric shapes that fit exactly into one another. And this is how Emil Fischer viewed the lock and key model, is that, for example, the enzyme serves as the lock while the substrate serves as the key. That the analogy between the interaction and combination of how a substrate fits into the lock of an enzyme is viewed as the lock and key model. In other words, that the specific type of enzyme will have the complementary shape of how the key is also shaped. This is how Emil Fischer looked at the enzyme and substrate in a lock and key model. The induced fit model is suggested by Daniel Koshland in 1958. Actually, this induced fit model is latest compared to the lock and key model that was proposed in 1894. And in this type of induced fit model, only the proper substrate is capable of inducing the proper alignment. In other words, it is not necessarily the complementary lock and key model. And when this is done, it will enable the enzyme to perform its catalytic function. So the induced fit model actually is a substrate that binds to an active site, and both can change the shape slightly, creating an ideal fit for catalysis to occur. So this is the induced fit model of Daniel Koshland, and you notice here that our substrate is not necessarily the equivalent to the lock and key model, but both of the substrate and the active site will complementarily fit to each other, and therefore the catalytic action can already proceed. So enzyme kinetics. The word kinetics simply means movement or motion. So enzyme kinetics is the study of the rate of enzyme catalytic reaction. There is an equation that is developed for the enzyme catalytic reaction, and this is by Michaelis-Menten model. So what is the equation about? According to the Michaelis-Menten model, that the initial velocity of the catalytic reaction is given by the formula. The maximum velocity multiplied by the substrate concentration in molar number divided by the substrate concentration at the half of its maximum velocity plus the substrate concentration. So that is the formula given by Michaelis-Menten model. An example to the rate of enzyme catalytic reaction is given by the carbonic anhydrase. This carbonic anhydrase catalysis rate is equal to 10 raised to the power of 6 of these carbon dioxide molecules in every second. This is also according to the Michaelis-Menten model. The rate of enzyme catalytic reaction is best in the range of pH of 5 to 9. Higher than 9 or lower than 5 can result into enzyme denaturation. In terms of the optimum temperature or operating temperature, enzyme is best between 35 to 45 degrees centigrade. Higher than 45 degrees centigrade can result to enzyme denaturation. Now, after eating, it is advisable to drink lukewarm water. This will facilitate our digestive enzyme as it is around 35 to 45 degrees centigrade to work best. So here are the different types of enzyme with their different optimal pH and optimal temperature or operating temperature. And in the last column is their maximum activity. And so for example, we have pepsin, the optimum or optimal pH is 2.5. So basically pepsin is operating in a very acidic environment, but it will denaturize after 40 or above 40 degrees centigrade. So when pepsin is exposed to a higher pH, pepsin will also denaturize. These enzyme kinetics play a big role in drug discovery. Why? Because enzyme kinetics reveal how drugs or agonists might inhibit the enzyme. And this also reveals how enzymes transform substance into products. Now, in the case of how drugs or agonists might inhibit an enzyme, an example to this is non-steroidal anti-inflammatory drugs, also known as NSAIDs. NSAIDs inhibit cyclooxygenases. This cyclooxygenases is actually an enzyme that is a mediator to inflammation and pain. And when cyclooxygenases enzyme is inhibited by this drug, we will almost entirely not anymore to feel pain. An example to this are aspirin, naproxen, and ibuprofen. Another example is the anti-cancer drug called methotrexate. Methotrexate inhibits the enzyme called tetrahydrofolate reductase. This type of enzyme is associated with the development of acute leukemias. When this enzyme is inhibited by this drug, it will no longer develop further into leukemia. Enzyme inhibition. Enzyme inhibition simply means that there is an inhibitor to stop a substrate from entering the enzyme's active site. The inhibitor that causes enzyme inhibition hinders the enzyme from catalyzing its reaction. So therefore, this requires enzyme inhibitor. Enzyme inhibitors are molecules that interact with the enzyme. Enzyme inhibition may be permanent, also known as irreversible, which is also known as enzyme poison. An example of the permanent irreversibility of the enzyme inhibition is the one with penicillin. Penicillin acts as an enzyme inhibitor because this inhibits the growth of the bacterial cell wall. So this penicillin functions by interfering with the synthesis of the cell wall of the producing bacteria. By this inhibition, an enzyme that is called transpeptidase is inhibited by the penicillin inhibitor. Another classification of enzyme inhibition is called the temporary or the reversible enzyme inhibition. The first type of a temporary or reversible enzyme inhibition is called the competitive inhibitor or competitive enzyme inhibition. The competitive enzyme inhibition or the inhibitor binds to the enzyme active site. So this is the enzyme, this is the active site, and this is the substrate. Once the substrate attaches to the enzyme active site, this becomes the enzyme and substrate complex, and the catalysis reaction can occur. But in a competitive inhibition, the inhibitor occupies the active site of the enzyme. Thus, the substrate can no longer take place to the enzyme active site, and the catalytic event of the enzyme or the biomolecular reaction can no longer take place. An example to this enzyme inhibition is that of statin or statin drug. The statin drug acts as the enzyme inhibitor. This is a statin tablet, also known as Lipitor. Statin, also known as Lipitor, inhibits the production of cholesterol. Another type of enzyme inhibition is called the uncompetitive enzyme inhibition. In an uncompetitive enzyme inhibition, the inhibitor binds only to the enzyme substrate complex. So this is how the uncompetitive inhibition works. The enzyme and substrate complex is where the uncompetitive inhibitor will bind. This is the normal binding between the substrate and the enzyme. An example to this uncompetitive inhibitor is the drug that is used to treat poisoning. The other one is called the mixed enzyme inhibition. This type of inhibition will only make the enzyme inactive for a while. Next type is the non-competitive enzyme inhibition. This binds equally to the enzyme and enzyme substrate complex. It is called a non-competitive inhibition because it do not compete with the substrate to its enzyme active site, but rather the inhibitor will be attached to the enzyme allosteric site. And this changes the conformation of the enzyme active site. The allosteric site is a site that allows molecules to either activate or inhibit or turn off or turn on the enzyme activity. The non-competitive enzyme inhibition only reduces the activity of the enzyme, similar to the mixed enzyme inhibition. An example to this is the heavy metal. Accumulation of heavy metal in the ground, for example, reduces or decreases the decomposition activity of the soil. Another example of a non-competitive inhibitor is cyanide. Cyanide suffocates the cell from getting oxygen. That's all for enzyme, and thank you for watching.